All right, so let's erase this and get started. All right, so the first one is moist heat sterilization. And I abbreviate a lot, so we're going to do that. It's easier and faster. So with moist heat sterilization, you're going to need to know, one, that involves boiling. Two, that involves autoclaving. So autoclaves, and that some viruses are destroyed. And then lastly, we know that endospores can be killed through moist heat sterilization. So let's stick with boiling. We'll start with boiling first. So with boiling, there are 3 microorganisms that you need to know that are killed or destroyed by boiling. One is fungi. Fungi and its spores. And its spores can be killed through boiling. And then viruses can be destroyed. And then our third one is bacterial pathogens. So, moist heat sterilization. In moist heat, we use boiling, this method of boiling. And microorganisms that are killed or destroyed are fungi, viruses, and bacterial pathogens. Remember that some viruses are destroyed using moist heat sterilization. Now with autoclaving, it's an instrument that is used for sterilization, sterilizing instruments, sterilizing dressings, solutions. What else? Culture media. So let's put that down here. Culture media. All right. Autoclave is an instrument used to sterilize certain things like instruments, dressings, solutions, and culture media. Now, using this instrument, we're going to need a very high temperature. So we need a high temperature and a high pressure. Now, endospores can be killed through by using autoclaves and through boiling. But it is through, the amount of time is 20 hours until they are destroyed. So that's moist heat sterilization. Now, if you can remember boiling and autoclaves. So with boiling, what microorganisms are killed? Then with autoclave, you need to remember that there is a high temperature and high pressure that is needed. And these are the what the autoclave is used to sterilize these particular things. And that you need to remember that endospores are killed through autoclaving and boiling. With boiling, 20 hours is needed to kill them. So let's switch up the color. We're going to do red and we're going to do dry heat sterilization. So with dry heat, we have 2 things that we need to remember. Flaming, and we're going to have to remember hot air sterilization. So hot air sterilization. So with flaming, we know that there is a direct contact with fire and the item that you want to sterilize. So it's a direct contact. And we see this example through or with our inoculating loop. So when we have a Bunsen burner and it's lit and we pass or hold our inoculating loop in the fire, we're going to see a red glow of the metal that allows all microbes that are on that loop to be destroyed. A method in flaming is called incineration. Now, with incineration, items that can be killed, items that can be destroyed, or contaminated items are dressings, cups, paper cups, bags, things like that. So our next one is hot air. So with hot air, the instrument used is an oven. And the temperature needed to sterilize is 170 degrees Celsius for the length of time of 2 hours. So what you'll need to do is remember flaming and hot air sterilization for dry heat sterilization. Remember those two things, then you can stem off and put important facts about flaming, important facts about hot air sterilization, about that subtopic. So dry heat is your main topic. Your subtopics are the flaming and hot air sterilization. From there, you put your important facts that will help you remember everything that you need to know about that subtopic. So our next one, let's do black. We're going to do pasteurization. So pasteurization. So with pasteurization, we have Pasteur. Oops, going too fast. Pasteur. We have our phosphatase test. Moving too fast. Phosphatase test. And we have HT, ST. And what else? Bacteria, thermoduric. Bacteria. So we have Pasteur, phosphatase test, thermoduric bacteria, HTST, and I will stop there. So these are pretty much the main things that you would need to know for pasteurization. So we're going to start with Pasteur. Now we know Pasteur was known for pasteurization, fermentation, but in this particular one, we know from history that he had used mild heat with alcohol or in alcohol. Now this mild heat was used to stop spoilage. That would mean that bacteria, or let's say better yet, microbes, were killed. Microbes that caused the alcohol to spoil. So he noticed that the preservation of the alcohol increased and the spoilage decreased when heat was used in alcohol. So with phosphatase tests, now this is a test that that's used to test in milk to see if the enzyme that's in milk. So it tests this enzyme, and this enzyme is in milk. If this test let's see, how should I say it? So the phosphatase test, phosphatase is in the milk. Now, the test is used to see whether the presence of this enzyme is there. And this will allow the scientists to know whether this milk has been pasteurized or not. So our next one is high heat or high temperature. So we have a high temperature, short time. And the temperature here is 72 degrees for the length of time of 15 seconds. Now, thermoduric bacteria, they are resistant to heat. They're heat resistant. So heat resistant bacteria. But they're not pathogenic. They won't cause harm to us or anyone who is consuming milk, and so no harm and no food spoilage. So that is pretty good. And then our next one, we can remember that pasteurization lowers microbial numbers, a plus, and then we know that it eliminates pathogenic, so eliminates pathogenic microbes. Eliminates pathogenic microbes. So, pasteurization. Remember that Pasteur is involved, that there is a test that scientists use to see whether phosphatase, the enzyme that's in milk, is present and whether the milk has been pasteurized or not. Another thing that you need to remember is high temperature, short time, and what that temperature is and what that time is. You can also know that there are some bacteria that are heat resistant when would not affect pasteurization of that milk. Like the bacteria will not cause any harm or spoilage. So pasteurization is important for lowering microbial numbers, and it eliminates bacterial microbes. So that's pasteurization. Our next one is sterilization. And let's do blue for that one. So this one's pretty simple and straight to the point. So one, sterilization involves UHT. Now UHT stands for ultra high temperature. So ultra high temperature. Now the temperature would be 140 degrees Celsius for the length of time of 4 seconds. See? Pretty quick. This is also used in, if you ever go to the restaurants that serve coffee and they have those little creamer cups. Sterilization using the ultra high temperature is used on those particular creamers. It allows the creamers to stay at room temperature without spoilage. So every time that you see those little cups of creamers, you can remember ultra high temperature. That treatment was used to sterilize that milk. Fun fact. You can also use this method or this treatment in sterilizing juice as well. So that's it for sterilization. And our last one is, Filtration. So we have filtration. And with filtration, it is used when we don't want to use the UHT method treatment or moist heat treatment or pasteurization. This is a no heat way of sterilizing things, and then I'll go into what we're going to sterilize. So these are heat sensitive items. Well, heat sensitive items or things. I shouldn't say items. Heat sensitive material. How about that? So we have culture media. We have enzymes, vaccines, and antibiotic solutions. Now in filtration, filters are needed, and we have here membrane filters. Now, some of these filters can be made up of plastic polymers or cellulose. Esters. So plastic polymers or cellulose esters. And another fact about the pores is that some of these filters can have a 0.01 micrometer filter pore size. This will allow us to capture, or scientists to capture viruses and or large molecules. If I can spell large molecules. How would I abbreviate large molecules? So with the esters, let's see, esters, cellulose esters, plastic polymers, for bacteria. So the pore size to capture bacteria are 0.22 micrometers and 0.45 microliters. So we have membrane filters that can capture viruses and bacteria. Another filter is called HEPA filter. And HEPA stands for high efficiency particulate air, and they are filters. Now with this particular filter, we have, we know that it can capture bacteria. Now it can only capture bacteria with an increase or a high pore size, higher than 0.3 micrometers in diameter. So microbes are removed. From the, let's say, removed from the antibiotic solution with a greater diameter size of 0.3 micrometers. And that is filtration. Those are the 5 points that I want to cover. So the next one I'm going to cover is questions, or questions that we need to answer to make sure that you remember everything that I just taught you. So let's see if I can find it. I think it's here and here. All right, we're going to bring that down. So here are the questions. So what moist heat sterilization method is best used to kill endospores in 15 minutes? And all of these questions, all of the answers that I have are in the lesson that I just taught. So you can always go back or rewind and get the answers from this video. And you put them in the comments. And don't forget to like and share this with anyone who is struggling with microbiology. That would be helpful to them. So the second question is, what is the intent of pasteurization? Number 3, non-refrigerated milk is treated under which treatment? High temperature, short time, or ultra high temperature? What is the temperature and length of time for hot air sterilization? And 5, what does HEPA stand for? Again, to find these answers, they are in the lesson that I just taught previously, and you can put your answers in the comments. I'll be in there checking your answers. And if you get them all right, I'll give you a shout out on my IG, which is at Microbiology Tutor on IG. All right, so we went over the lesson and we have our questions, and you're going to be answering them in the comments. I'll be checking. The next one is micro mind map. So what you saw previously from what I did, let's exit out so you can see here. So what you see here is micro mind mapping. I have our main topic, moist heat sterilization. That's our main topic. And from there, I stemmed out to subtopics, boiling and autoclaving and endospores. Those are my subtopics. After that, I stemmed out from those subtopics into smaller details about that subtopic. So the first thing is to get the core. You have your main topic, and then you have your subtopics. So once you get that, you're halfway there. To remember the particular things that you need to know, like for your exams, then you stem out to these details. Now, if you want a detailed outline or guide to micro mind mapping, I do have a microbiology survival guide, and I'll put it in the description after this lesson to teach you how to micro mind map. It's an easy way for you to extract all of the important information that you need to know about microbiology without writing extremely long outlines. I think this is very quick and easy and simple to do. So with the micro dumping section right after the questions, you would practice this mind mapping until you can get it without using your notes. So that is the third one. And the next one is, guess what? We're going to have fun with micro. And how are you going to have fun with micro? By testing your knowledge. So I have interactive slides that would help you remember microbiology, kind of take a break from reading the textbook. And so this one is, what physical method is used here to control microbial growth? And here I have the little facts about the particular sterilization method. So this one kills by oxidation effects. This method is used to sterilize inoculating loops. Items are sterilized using an oven. And out of that, you have to pick which one is the correct one. And I'm not going to answer it because I know that you guys can, and you can also put the answers in the comments. So after that, if you get the right answer, you're going to see correct. If you get the wrong answer, you're going to see wrong, and it will take you back to the original slide. So the next one, it kills some viruses, fungi, and its spores and bacterial pathogens by boiling. Autoclaves are used. Endospores can be eliminated by autoclaving and long boiling periods. So here you have to pick which one is the correct answer. If it's correct, you'll see correct. If you see wrong, then you're going to go back to your original question and try again. UHT, ultra high temperature treatment at 140 degrees Celsius for 4 seconds. Which one is the correct answer? Again, you can also put it in the comments. Let me know what the answer is. Correct or not correct? Next one, used for heat sensitive materials, membrane filters, HEPA filters. Which one is the correct answer? Correct or wrong? Pasteur added mild heating to alcohol. HT, high temperature short time, 72 degrees Celsius for 15 seconds. Phosphatase test and eliminates pathogenic microbes. Which one is the correct answer? Correct or wrong? So once you have you've completed this interactive slides, so we went over a lesson and we did our practice questions. We did a micro mind map within that lesson, but I do have a guide for you, and we went over test your knowledge interactive slides. So in replacing the interactive slides, I also do games, and I do Wheel of Microbiology and Micro Jeopardy. I play some of those games on my IG, again at Microbiology Tutor on my IG, and those are pretty fun games to break up reading your texts or watching videos. It allows you to have fun with micro and also absorb the information to retain the information. So if you are interested in having a study session, just like me being with you, let's go back. And there you go. So with me being with you tutoring, but through video, I have that available for you. So it's an first one that you'll get is an instructional video. I show you how to study microbiology, how to study, how to conduct these study sessions. And then I also have lesson videos. I teach you the topics about that's in microbiology. I have practice foundation questions like you've seen here in this video, questions that I've created from the textbook that will help guide you through your chapters. I have a guide to help you micro mind map, and that's dumping what you've learned through the lessons, through the questions, and dumping everything that you know on paper. So if you can explain it on your in your own words, then you can then you know the material. And that's what a micro dump is. It helps you get that information out on paper and allows you to explain it. And then the last one is test your knowledge interactive slides, what I just covered previously. And or you can also have games that you can do. So that is what I have for my study sessions. If you are interested in having this complete session wrapped up in a box with a little bow on it, I will be more than happy to create that for you. You won't have to search for videos on that particular topic. It'll be all in one module. You won't have to search for questions. It'll all be in one module. The micro mind mapping guide, the guide will be in the module, and testing your knowledge interactive slides will be in that module all in one. So it's no more wasting time. No more wondering what you're going to do for your study time. It's all done for you. All you have to do is click on the module, open it up, and study. All right, thanks for joining me on this live. I had so much fun, and I can't wait to see you guys in the next one. It will be next week, next Friday, same place. The topic will be environmental microbiology. So that's what I'll cover for the week. Don't forget to like this video, like my previous video that I made this week. Comment and put your answers in the comments. I will check them, and if you get them all right, I'll give you a shout out here on YouTube and on Instagram. What else do I want you to know? I think that is it. Like, comment, subscribe. That would help. If you would like, feel free to subscribe and hit that bell to be notified of more videos that I will upload next week. Environmental microbiology.